Hello everyone, I'm MS Shweta from 2nd MSc Microbiology. Today I'm going to present the topic xenobiotics. Xenobiotics is a foreign chemical substance found within an organism that is not normally or naturally produced or expected to present within. Basically, xenobiotics are a chemical compound which are uptaken by our diet foods or other chemical substance. It can also cover substances that are present in much higher concentration than are usual. Specifically, drugs such as antibiotics or xenobiotics in humans because the human body does not produce them itself. Are they a part of a normal food? Biological effects of xenobiotics. Xenobiotics can produce a variety of biological effects, including pharmacological responses, toxicity, immunological responses, and also it may cause cancer. Types of xenobiotics: exogenous and endogenous. In exogenous, the foreign molecules which are not normally ingested are utilized by the organisms, but they can enter through dietary foods or in the form of certain medicines or drugs used for the therapeutic causes or inhaled through environment, such as drugs, food additive, pollutants, insecticides, and carcinogens. Endogenous. Though they are not a foreign substance but have effects similar to exogenous xenobiotics. These are synthesized in the body or produced as a metabolisms of various processes in the body, such as bilirubin, bile acids, steroids, and fatty acids. Metabolism of xenobiotics. Metabolism of xenobiotics occur in two phases. Phase 1. The major reaction involved in hydroxylation. In addition to hydroxylation, a wide range of reactions also takes place including deamination, dehalogenation, desulfuration, reduction, peroxygenation. Deamination means the removal of amino group from molecule. Dehalogenation means chemical reaction involved in cleavage of carbon-halogen bonds. Desulfuration means removal of sulfur compounds from the molecule. Reduction: transfer of electrons from one to another molecule. Peroxygenation means replacing nitrogen in the alveolus with oxygen. In phase 2, the hydroxylate or other compounds produced in phase 1 are converted by specific enzymes to various polar metabolites by conjugations with glucuronic acid, sulfate, acetate, glutathione, amino acids, and also by the method of methylation. Structural features of xenobiotic compounds. Instead of hydrogen, halogen is present in the molecules which needs additional energy for cleavage. Other groups like sulfonate, nitro group, amino group, methoxy, etc. are also present in the xenobiotics. Cycloalkanes, aromatic compounds, and heterocyclic compounds are more reluctant. Branched linear chains are resistant to biodegradation. Halocarbons. These xenobiotic compounds contain halogens in diverse amount, such as chlorine, bromine, fluorine, or iodine instead of hydrogen atoms. They find their applications in various ways, such as propellants in spray cans of cosmetics, paints, etc., and solvents such as chloroform. They are also used in condenser unit of cooling system. There are some examples: Furans, CC113F, CC12F2, etc., and as insecticides DDT, BHC, linzane, etc., and herbicides such as dalafon chloroform and furans, which are basically volatile in nature and released into the atmosphere where they devastate protective ozone layer that leads to increased UV radiation. Second one, polychlorinated biphenyls. They are basically utilized as coolants in transformers or as exchange fluids for heat. Third, synthetic polymers. These are polyethylene, polyvinyl chloride, polyester, etc., and nylons, which are used as article of clothing enveloping material. Alkyl benzene sulfonates. They are surface active detergents that are superior compared to soaps. The sulfonate groups at one end opposes the microbial degradation. Fifth one, oil mixtures. Oil is miserable in nature, basically because it is not soluble in water. And due to the lethal nature of some of its compounds, biodegradation is used when there is a small of soil seepage. But when large spill occurs, these complexes are reluctant in nature. Other xenobiotics. Compounds containing aliphatic cyclic ring arrangements that contains nitro, sulfonates, amino, methoxy or carbamyl groups other than halogen groups. These properties make them more reluctant. Effects of xenobiotics. Metabolism of xenobiotic can result in cell injury, immunological damage, or cancer. Cell injury, that means cell toxicity, can be severe enough to result in cell death. These macromolecular targets include DNA, RNA, and also protein. The reactive species of a xenobiotic may bind to a protein, alternating its antigenicity. The resulting antibiotics can then damage the cell by several immunologic mechanisms that grossly perturb normal cellular biological processes. Reactions of activated species of chemical carcinogens with DNA are of the great importance in chemical carcinogenesis process. Thank you for listening.